Hello, and thank you for joining us today. My name is Zoe Etter, and on behalf of MEGIN, I'd like to welcome you all to this MEGIN Masterclass with our guest speaker, Professor Peter Uhlhaas. Please use the Q&A button on your screen to send in questions during the webinar. There'll be time to answer your questions at the end of the session. This webinar will also be available to watch on demand on the MEGIN website at a later date. So without further ado, it gives me great pleasure to hand over to our speaker for today, Professor Peter Uhlhaas, who will officially start the webinar. Thank you.
Okay, thank you so much for the invitation and the opportunity to present our work, which is going to be on MEG in translational neuroscience, present and future applications. During my lecture, I'm going to cover 4 topics. I'll provide an overview and rationale for the use of MEG in translational neuroscience. In the second part, I'll focus on the role of neural oscillations in schizophrenia and the contribution of EI balance parameters and how we can assess these with MEG. In the third section, I will talk about work that we have conducted to identify MEG-based biomarkers for early detection and diagnosis of psychosis. And I'll try to provide a summary with a brief outlook how we are going to move things forward. Okay, so this is not much news to you. MEG is a tool that can be used to measure the deviations within a magnetic field that are created by electric currents of the brain. It was first developed in the 1970s, and first commercial systems were roughly available in the 1990s. And while MEG systems have spread and developed, it's probably fair to say that the clinical applications have been somewhat limited and they're largely confined to epilepsy in certain parts of the world. And therefore, MEG continues to be mainly used in basic research. MEG has a number of, I think, important properties which make it important for translational neuroscience. One, of course, is its excellent temporal resolution, the fact that we have also a very good spatial resolution, at least for electrophysiological techniques. And also importantly, it gives us a direct measure of brain activity, which is amenable to mechanistic interpretations. And that's particularly important if we're trying to identify the origins of certain disorders. So mental disorders, as most of you are aware of, are and continue to be a leading cause of non-terminal illnesses. And while we have a range of pharmacological and psychological interventions available, overall they probably only have modest efficacy in terms of the effect sizes, and also the large majority of patients do respond, but there's also a significant proportion of patients who will not basically improve. Also, what we are lacking are essentially biomarkers, so that would help us, for example, to identify certain treatments, and also to allow maybe early detection of certain cases, early diagnosis. And that is basically the question what I'm going to address: to what extent MEG might be useful for such purposes. So to what extent can MEG, for example, be useful for early detection of, for example, schizophrenia? So I'd like to continue with a brief definition of what a biomarker is. So that's the definition from an NIH working group, which basically provides a, I think, good working definition. So here the emphasis on a objectively measured process that can be indicative of a certain pathophysiology, pharmacological response, or also a diagnostic marker. You see here also the overview of different types of biomarkers. So we can, for example, have predictive biomarkers in certain at-risk samples. We can have, for example, also monitoring biomarkers. In my presentation, I'm going to mainly look at the possibility that MEG is used, for example, for prediction of certain disorders, to establish whether somebody is at risk, maybe also as a diagnostic biomarker. And I'm also going to show some data that suggest that MEG is very sensitive to certain pharmacological manipulations. So these are the aspects that I'm going to focus on. So in my talk, I'm going to mainly focus on schizophrenia. This is not meant to say that MEG has only applications to this disorder, but this is probably where a certain amount of work has been done, and certainly in my group, we've basically used MEG for probably now more than roughly 15 years, I would say, to study its role in trying to understand what the origins of schizophrenia are. Most of you will be familiar with these facts. So schizophrenia is actually a fairly prevalent disorder with roughly 1% of the population being affected. And what's probably most important is that that still for the large majority of patients, we continue to have a poor outcome in the majority of patients. So that means that only 10 to 20% of patients will actually make a full recovery. And one of the reasons for this poor outcome is the fact that schizophrenia, like the majority of mental disorders, emerges during the transition from late adolescence to adulthood and interferes quite profoundly with the important ongoing developmental processes of this period. And that basically leads to the very pronounced deficits in terms of functioning and so forth. And probably, we also have to here summarize that there have been largely no significant advances in both psychological and pharmacological approaches in the treatment of schizophrenia, except for a recent development of a new muscarinic pharmacological approach, which is, I think, very exciting. But overall, the outcomes, and this is supported by recent data, haven't really dramatically improved. So there is still a lot of work to be done in trying to understand the origins of this disorder. And the hypothesis that we and many other people are pursuing is that schizophrenia, maybe also other syndromes, we can maybe discuss this later, are related to disturbances in the balance between excitation and inhibition that is on the one hand manifested also in disturbances in neural oscillations. And in my talk, I'm going to particularly focus on the higher frequency range, so-called gamma band oscillations, which are roughly oscillations between 30 and 100 hertz. And one of the reasons for this focus is that firstly, they have a prominent role in brain functioning and in bringing about coordinated action and cognition. And secondly, we have some well-established findings from basic neuroscience that basically highlight that certain circuit motifs, for example, specific GABAergic interneurons, but also their excitatory drive, are mechanistically relevant for the generation of these high frequency oscillations. And that basically brings us closer to linking, for example, MEG signals to underlying circuit dysfunctions. And I think this is a very important aspect if you want to firstly understand the causes of such a disorder, and secondly, also then develop maybe treatments based on these insights. So what you see here are firstly some data from post-mortem studies of schizophrenia, which show the alterations in GABAergic interneuron markers in schizophrenia. And the reason why I'm showing you this is that firstly, to demonstrate that different interneurons are affected in schizophrenia. So here these columns show you the degree of reduction, for example, here in red, somatostatin interneurons, and in orange, for example, parvalbumin interneurons. And secondly, that this is basically something that is present across different cortical areas. So for example, here we can see that these reductions in both somatostatin and parvalbumin are present in the prefrontal cortex, but also importantly in sensory areas here, for example, in the primary visual cortex. And this is quite relevant because I'm going to show you mainly data from sensory paradigms, also from listening state activity measured with MEG. And that basically provides the motivation for basically studying these sensory processes. And secondly, you see here some patient quotes that describe very prominently alterations in visual perception in patients with schizophrenia, here also in auditory perception, that highlight that the sensory abnormalities are a very important aspect of schizophrenia, and this therefore might provide a good starting ground for trying to understand these deficits. One paradigm that we've used quite a lot, and I'm going to show you several data sets of, is the so-called 40 Hertz auditory steady state paradigms. So basically, steady state paradigms involve the presentation of a temporally modulated stimulus, and what can be observed is that the brain basically entrains to the frequency of that stimulation. So this can be demonstrated in vision, but also here during auditory perception. And there's an interesting aspect towards auditory steady state responses in the sense that the peak frequency, so you might refer to this as the preferred frequency of auditory cortices, is at 40 Hertz. So that is where the maximum amplitude in response to these auditory steady state stimuli can be observed. And you see here some example data from both EEG and MEG, where we can see basically the corresponding time frequency activity. And not surprisingly, we can basically observe here that both in EEG and MEG, we see this sort of sustained 40 Hertz response in relationship to a 40 Hertz auditory state stimulus. What is interesting here is that if you look at the topography at sensor level, that in EEG you find that this is much centrally located, and we have a much better spatial resolution at sensor level here in MEG data. Schizophrenia patients show pronounced deficits in both the spectral power and what we refer to as the intertrial phase coherence in regards to the 40 Hertz response. So this is a figure from a meta-analysis that we published some years ago, suggesting that this is a fairly robust finding. So we have reductions with medium to large effect sizes, both in terms of spectral power and also intertrial phase coherence. And what is also interesting and relevant for some later findings is that the entrainment is not something that you can observe only in auditory cortices, but it seems to propagate much beyond that, such that we can actually find generators, for example, in subcortical regions, such as the thalamus and the hippocampus. So this is potentially a very useful paradigm to probe circuit dysfunctions. I also would like to show you some pharmacological data that can actually demonstrate that these oscillations are sensitive towards pharmacological manipulation, suggesting that they closely reflect EI balance parameters. So this is data from the lab of Tobias Donner, with whom we collaborated on this finding. So here, Toby's lab examined on the one hand lorazepam, so which is a GABA alpha receptor agonist, and memantine, which is an NMDA receptor blocker, and they investigated to what extent both the GABAergic and glutamatergic neurotransmission would impact on the 40 Hertz auditory steady state response. And basically what we can see here is that there are interesting differences, in particular in regards to the effect of lorazepam, where we can see here in the blue trace that the sustained 40 Hertz response is significantly elevated through the enhanced GABAergic neurotransmission relative to the placebo condition. And in this example here, the memantine doesn't seem to have a very pronounced effect, suggesting at first that GABAergic neurotransmission can be closely investigated with this paradigm. And I should also note that these are MEG data, so very beautiful, demonstrating that these parameters are very sensitive towards certain pharmacological manipulations. I'd like to briefly continue this with data from rats, where similarly here, 40 Hertz auditory steady state paradigm was applied during a ketamine challenge here at different dosages. And what you can observe here is that at lower dosages, you see here in terms of the intertrial phase coherence, a significant increase in terms of the phase consistency in the gamma frequency range, obviously, and at higher dosages, there seems to be a reversal, whereby ketamine seems to actually reduce the 40 Hertz ASSR response, but I think quite beautifully highlighting that also glutamatergic neurotransmission is closely modulating these responses. And this is important because we think that both of these neurotransmitters are relevant for understanding some of these circuit deficits in schizophrenia. I would like to continue with another data set that shows, I think, quite nicely the strong sensitivity of MEG signals towards pharmacological manipulation. So here we also administered ketamine to healthy volunteers, and ketamine is interesting because ketamine on the one hand elicits certain cognitive and psychotic symptoms in healthy volunteers, and also we know that the NMDA receptor, which is basically reduced in activity through ketamine, basically is significantly implicated in schizophrenia. Therefore, trying to compare and modulate the gamma activity through ketamine can maybe also tell us something about the pathophysiology of schizophrenia. So this is what we've done here. This is from a slightly older data set where we administered ketamine in a within subject design in healthy volunteers and applied a bolus of ketamine followed by a constant infusion. And below you see the resting state activity in healthy controls, showing the difference in gamma band power on the left two figures compared to placebo. And I think what is quite remarkable is the size of the gamma band power increase. So ketamine seems to drive up the power frequency in between roughly 30 and 80 Hertz. And this can also be localized. And interestingly, we observed here that the maximum of the ketamine related increase was in subcortical areas, especially in the hippocampus and also in the thalamus. And this is relevant because these regions are also very rich in NMDA receptors. We've recently followed this up, and we also had a slightly closer look at the MEG activity. And the reason why I'm going to show you this is that I would like to basically show you that if you look at the single subjects, which you can see here, so these are individual gamma band power values in placebo on the left, and this is the ketamine condition, you see that on a single subject level that ketamine drives up highly robustly the power in gamma band frequency ranges, which I think again shows that MEG is highly sensitive to these pharmacological manipulations, even at single subject levels. And I think this is something quite important to demonstrate. In the next step, we were also interested in trying to understand what might be the mechanisms of these ketamine-induced effects. So it should be noted that ketamine does not only act upon glutamatergic neurotransmitters, it has a wide range of activity, but it's assumed that the main psychomimetic effects of ketamine are due to the blockade of the NMDA receptor. And here we ask the question, what are the interneurons that are predominantly affected by ketamine? So we know that ketamine has, or NMDA receptors are present on different types of interneurons, and you might remember that on the one hand we have the paldalbin expressing interneurons, somatostatin expressing interneurons that are reduced in schizophrenia, and also here we have a third class, so-called vasoactive intestinal peptide interneurons. And these are associated with slightly different firing properties that are related also to the different frequencies they have been involved in. So here we were wondering, can we use computational modeling to maybe figure out which interneurons and their NMDA receptor drive might be predominantly related towards this gamma band dysregulation following ketamine. So here we made use of a very specific and detailed circuit model that targets here layers 2, 3, and then basically systematically manipulated different GABAergic interneurons and the NMDA receptor drive In addition, we also looked at the effects on pyramidal neurons to also replicate the increased excitatory drive and compared these data to the actual MEG data in order to basically find out which GABAergic interneurons might be preferentially targeted for NMDA receptors. So what you see now here are the simulation results for different reductions of NMDA receptors on the PV interneurons on the left, the somatostatin interneurons in the middle, and the VIP neurons on the right. And basically what we can observe here is that both PV and the so-called somatostatin interneurons show a significant relationship whereby reductions in both PV, so NMDA receptor expression and drive on PV and somatostatin interneurons leads to this upregulated gamma activity, which is not the case in the VIP interneurons. So that suggests that the NMDA receptor blockade can very selectively target interneurons, and the resulting disinhibition could be a potential mechanism for the emergence of these dysregulated gamma band oscillations, which is also relevant for understanding schizophrenia, because we can all sometimes observe alterations in spontaneous gamma band activity. But I think also this shows very nicely how we can combine MEG data with computation modeling in order to understand the underlying mechanism of signal changes. In the next few slides, I would like to slightly shift gears and highlight another important aspect of schizophrenia, which is the fact that schizophrenia can be conceptualized what we refer to as a disconnectivity syndrome, so that somehow the cognitive deficits, which are very prominent, but also certain symptoms of psychosis, such hallucinations and delusions, may derive from aberrant functional interactions. So this was already proposed more than 100 years ago, and more recently was captured, for example, in the formulation of Carl Friston and Chris Riff, and then description of a disconnectivity syndrome. And what I would like to show you briefly here is a way of using MEG to quantify such disconnectivity patterns. And this is from a study we did a few years ago together with Sato Matthias Palva. So here we made use of a data set that we still obtained in Frankfurt, where we used more complex visual images, so-called munifaces, which you see here on the left. So munifaces are photographic images of human faces where all shades of gray are removed, and as a result you see this sort of fragmentary depictions of human faces. And the theory goes that in order to make out that you see a face here, you have to visually close the missing information. And what also has been demonstrated previously, that if you invert these stimuli, and this is very consistent with older face perception data, that no face can be perceived. And this is also a process that patients with schizophrenia find quite difficult in terms of grouping of visual information in coherent object representations. And we studied this here in a sample of chronic schizophrenia patients. So they basically were presented with a set of these munifaces that were presented either upright or inverted. And we first looked at the behavioral data, and basically what we could observe here is that schizophrenia patients were significantly impaired in the detection of upright munifaces, but were largely intact in response to the inverted stimuli. The probably more relevant point to this audience is here then the resulting analysis, because here we examined phase synchronization, which of course can be also examined in different ways. So I've early on mentioned intertrial phase coherence. So that's a measure of phase consistency for single channels. Here we were interested in examining the question to what extent long-range synchronization might be critically implicated in the disconnectivity syndrome, and therefore we computed phase locking across all possible source space areas, and I think probably for the first time provided a sort of MEG-based layout of functional disconnectivity in source space, which you can see here in the top right. So these are the connections that show significantly increased phase synchronization, here in particular in the gamma band range between frontal visual areas, but obviously also across the hemispheres, which I think show quite beautifully sort of the widespread patterns of synchrony that emerge when these coherent object representations emerge. Of course, the primary goal of this study was to see to what extent this large-scale phase synchronization might be disrupted. So again, providing a sort of physiological substrate for a disconnectivity syndrome. And this is what we can see in this plot. So here, basically, we computed a difference map for responses to upright versus inverted phases in both clinical groups, and then compared these statistically. And what we can see is that in the schizophrenia patients, there was a significant reduction relative to controls when they basically generated these oscillatory patterns for upright versus inverted phases, around 200 to 300 milliseconds, which most people would agree with would be critical for the generation of coherent object representations. And this also occurred again in the 40 Hertz range, so suggesting that there might be a failure in large-scale synchronization that could drive some of these perceptual cognitive processes. So this is an example how we can harness MEG to basically physiologically, realistically depict such disconnectivity syndromes. So we're now coming to a third part, and that addresses the question to what extent MEG can be used potentially for early detection and diagnosis of psychosis. So on the left here, you see a figure that shows the different stages of what we refer to as psychosis. So psychosis basically is a wider term for describing mental experiences that involve a loss of a reduced contact with reality. So schizophrenia is a psychotic disorder, but psychotic symptoms such as delusions and hallucinations can occur across different psychotic disorders and sometimes also in affective disorders. And basically, more than 20 years ago, the idea was proposed that, well, perhaps what we could try and do is we can maybe detect young people who might have a high risk for psychosis and see where we can maybe predict the actual onset of what we would refer to as a first episode. Because if we're successful in this, this could basically open up the way to maybe also trying to intervene prior to the onset of psychosis and schizophrenia, and possibly even prevent the occurrence of a disorder in the first place. That's, of course, a very ambitious goal. And in the next few slides, I'm going to show you basically the approach, which is based largely on clinical signs and symptoms. So basically, scientists and clinicians have identified a number of different symptoms that seem to be characteristic of this, what we refer to as a clinical high risk for psychosis state. So basically, certain clinical symptoms, in particular, what we refer to as attenuated psychotic symptoms. So attenuated means psychotic symptoms reduced in intensity and frequency, seem to be a significant predictor for the later transition to a full-blown psychosis. And that has, I think, gained a lot of traction and interest because of the possibility to potentially prevent the onset of psychosis. And but you can maybe already guess that, you know, this population of at-risk participants is highly heterogeneous, and therefore it's important to develop biomarkers that could potentially tell you whether a young individual might be truly at risk for psychosis or not. I've brought you here a brief case note of a patient so that you can maybe get an idea as to the sort of clinical presentation. So this is Aaron, a 17-year-old high school junior who was referred for an evaluation in a university-based research program for individuals at clinical high risk for psychosis. He had undergone 3 brief inpatient stays prompted by his father's concern about his suspicion and unusual behaviors and his sudden decline in academic performance. He reported that almost every day he wondered if what he was watching on television is just a message from me. He had also wondered whether people knew things about him, even though they could not read his mind. At times he wondered if he might not exist, or he asked himself, is this real, or are we all in pods? In the interview, he linked the notion of pods with a film Inception, in which the characters move through shared dream worlds. Notably, Aaron was able to spontaneously question his conviction in these thoughts, and he noted that he always had an active imagination. So the latter point is very important because this basically would tell us that he is not yet fully psychotic. So he still is what we would refer to as able to reality test. But you can already see that some of these symptoms are rather bizarre. So if we were, for example, seeing such a person, we would certainly think that, you know, they may have a significant risk for psychosis, and we would like to continue basically seeing them. So what happens with these young people who display similar symptoms? So basically, this is an overview of the risk of psychosis in these young populations. And basically, the main point to take across from this slide here is that with increasing follow-up time, basically the risk of transition increases, maybe not surprisingly. And what we have currently is roughly 20%, in particular those with so-called attenuated psychotic symptoms, so this is also what Aaron presented, have roughly a 20 to 25% risk of developing psychosis within a 2 or 3 year period. Okay, so you might think this is quite good, but you might also think, hey, you know, you need to do better if you want to intervene in such individuals, and therefore, basically, we and others have gone onto the quest of trying to see what we can basically predict or specify whether somebody is truly at risk or not. I'm going to show you some data from a study we have set up quite a while ago, still at the University of Glasgow, where we recruited a large sample of these clinical high risk for psychosis individuals. Also, we compared them with so-called CHR negative, so a psychiatric control group, healthy controls, obviously, and a first episode psychosis group. And then we conducted comprehensive baseline neuroimaging measurements to address the question whether we can potentially predict the onset of psychosis in these individuals. You see that in addition to MEG, we also obtained MRS, some structural data, but of course, for the audience here, I'm going to focus on MEG and the question to what extent MEG might be predictive for the development of psychosis. So coming back to our auditory steady state paradigm, so here presented again amplitude modulated tones that were sometimes interspersed with a flat tone to engage attention. And here we have 4 different groups. So on the one hand, we have our clinical high risk for psychosis group, which consists of roughly 116 participants. We had a group of first episode psychosis individuals, a group of psychiatric controls. So these went through the same screening pathways as our psychosis risk participants, but they didn't meet the same criteria. And we also had healthy controls. So let's focus first on this part of the figure here, where we can basically see a bandpass filtered MEG signal in response to this 40 Hertz auditory steady state stimulation. And here we have 4 different groups. We have in red the first episode group, so this is a group with manifest psychosis. We have our at risk for psychosis group, which is here in blue. We have here in green our clinical high risks, which do not meet psychosis criteria, and our healthy controls. And what you can see is that there are significant differences in the amplitude fluctuations, in particular in the clinical high risk for psychosis and the first episode group. And you see that they're very closely together, so to speak, in terms of their decrease in amplitude, suggesting that their ability to follow or train towards this 40 Hertz stimulus in terms of the amplitude is already significantly decreased. So we followed this up through further analysis because, of course, we were interested to see which brain areas might be involved in this process. And what you see here are beamforming results where we can show that in addition to the auditory cortex, there was a significant modulation across subjects, also in the superior temporal cortex, and also the hippocampus and thalamus. So again, highlighting that this is a network phenomenon. And then, of course, we followed up these individuals to determine whether these signals might be predictive for the clinical trajectory. So coming further to the regions of interest analysis. So here we have three different regions where we compared our clinical high risks with first episode patients and healthy controls and the psychiatric controls. And I may just briefly explain again the color coding. So in red, we have our first episode psychosis group. In blue, we have our clinical high risk for psychosis group. In green, we have our psychiatric controls. And these are now displayed here in comparison to the healthy control. And therefore, a lower value here suggests a lower response in the right Heschel's gyrus, right thalamus, right hippocampus. And basically what we can see is that in terms of the amplitude, there's a strong overlap in terms of the reduction in 40 Hertz entrainment power in both the first episodes and the clinical high risk for psychosis individuals, which is present in both the thalamus and the hippocampus. There's also a reduction here in the right Heschel's gyrus, but this is somewhat more significant in the first episode psychosis group. And what we can also see that in terms of intertrial phase coherence, the clinical high risks have also a significant impairment in the right Heschel's gyrus. So this is already quite interesting. So we can demonstrate that already at baseline, there seems to be a significant impairment in both the FEPs and the clinical high risk for psychosis individuals. So potentially you could conceptualize this as a sort of diagnostic biomarker, which is fairly specific because we don't see this in the psychiatric controls. And now we're coming to a sort of core question, can we actually predict the clinical trajectory? So in order to look at this question, we have to again to adopt a different color code. So now we're comparing the different subgroups of our clinical high risk for psychosis individuals. So let me guide you through this. So in purple, we have our clinical high risk for psychosis individuals who developed a first episode of psychosis during our follow-up time. In red, we have our clinical high risk for psychosis individuals who continued to meet clinical high risk criteria after 12 months. In green, we have our basically remitted clinical high risk for psychosis individuals. So basically, these did not show at follow-up clinical high risk symptoms anymore. So they became, quote unquote, a bit more normal. And here in yellow, we have our psychiatric controls. And again, this is plotted compared to the healthy controls. And basically what we can see here is that the clinical high risks who transitioned to psychosis showed the strongest impairment, particularly in the right thalamus and the right hippocampus in terms of the spectral power entrainment, closely followed by those clinical high risks who remained at risk, so to speak. And this was significantly different from the remitted at risks and the psychiatric controls. We see a somewhat similar pattern in the ITPC and right Heschel's gyrus, again, showing that the transition cases showed the strongest impairment, but we also saw here, interestingly, an impairment in the remitted at risks. So that suggests that indeed, perhaps these 40 Hertz ASSR responses might be prognostic, so we can actually predict the clinical trajectory. And we followed this up now with a machine learning approach where we basically generated AUC curves and looked at the resulting model. And here you see the main results. So if we classified our at-risk participants into those with persistent versus non-persistent APS, so this is the comparison between those at risks who continue to meet at-risk criteria and the sort of healthy remitted at-risk individuals, we could very accurately classify individuals into these two groups. This was also statistically significant in the comparison for the transitioned CHRPs, so clinical high risk for psychosis, who developed a first episode versus those who did not transition. But here you see the statistical model, which was largely driven by the activity in the right thalamus, was significant but not quite as robust, and this is in some degree related to the smaller sample size. So this is something that we have to look at a little bit carefully, but nonetheless, the model was significant. In this context, again, coming briefly to some computational work that is ongoing in our group, I think that computational models, hold quite a bit of promise in order trying to understand mechanistic hypotheses. So here we're currently using a realistic biophysical model of the auditory cortex, which was generated by Durrell Bernal, working at SUNY New York. And basically what this model allows you to do is to manipulate both layer-specific and also interneuron specific different glutamatergic neurotransmitters in order to see whether the simulated data basically matches what you observe, for example, in your MEG findings. And that provides, for example, a very, powerful avenue in order to, again, try to arrive at some of the fundamental questions in schizophrenia, such as, for example, are these deficits primarily generated by deficits in GABAergic interneurons, and if so, in which subtypes, as opposed to, for example, a secondary response in excitatory drive. So this is ongoing work, but again, I wanted to highlight this in terms of the, promise of computational approaches to, model MEG data and basically also use this to understand circuit deficits, which I think is extremely important if we want to try to understand the origins, and I think in the medium long run to develop treatments based on this. I just would like to briefly highlight that we cannot only predict clinical outcomes in these at-risk individuals with auditory steady-state responses. We can also do this with a visual paradigm that we've published. This work was also led by Tnik Ren Tiong, where we can show that if we use such a visual grading, in particular, the first episode cases are impaired. And here we found that, in particular, the intertrial phase coherence elicited by this visual grading responses seem to be prognostic also for the clinical high-risk individuals. I'll keep this a bit shorter because we're coming towards the end of the presentation. And I just would like to basically highlight the main points, which I hopefully was able to communicate to you in a meaningful and coherent manner. So I've shown that deficits in gamma band oscillations are an important aspect of the pathophysiological architecture, let's call it, in schizophrenia, and which is consistent with impairments in EI balance parameters. And I think, the data that you were able to see shows that MEG measured gamma band oscillations are very sensitive to EI balance alterations elicited by pharmacological manipulations. And I've also shown you some first data that MEG might also be harnessed to predict the onset of psychosis in CHRPs. So indeed, MEG could constitute a prognostic biomarker. And I've given you also some examples how computational modeling can be used to understand also the basically underlying neurobiological mechanisms of certain signal changes. And I think this is, going to be important for the future. Maybe just one further outlook, how we are currently continuing this work. So while being a MEG enthusiast, I'm also aware of certain limitations, at least with traditional MEG systems. So we are currently working on new OPM MEG systems, which have been spearheaded by Matthew Brooks and others. And I think they have shown quite a bit of promise in maybe addressing some bottlenecks of traditional squid-based systems in terms of the availability of MEG systems. And so we are currently doing a first study using OPM MEG in schizophrenia patients. And also we are currently setting up a new OPM MEG center at Charité together with colleagues from the German Metrological Institute. And we hope that this can move this field forward, and in particular in the application towards translational neuroscience research. So on that note, I would like to acknowledge the people who did the work, and as you can see, this has spent many different labs. So the work on clinical high risk for individual psychosis individuals was largely conducted at University of Glasgow, with many people involved, particularly Tineke Grant-Young, Matthias and Satu Palva. And the current work on OPMs is collaboration with the German Methodological Institute. And here are the funding bodies, in particular the MRC, which I would like to acknowledge. And I would like to thank you for your attention. Thank you.
Thanks very much for such an insightful talk, Professor Uhlhaas. We now have time for some questions from the audience. Please feel free to leave your questions in the chat as we go along. So the first question, is there an interest for higher frequencies brain oscillations than 40 or 70 hertz in schizophrenia? And if yes, in which frequency or frequency bands?
So people have played around with auditory steady state responses also at 80 hertz, and I think there's some impairments also detectable at this frequency ranges. But I probably would say that you get more signal at 40 hertz. So I think, you know, while interestingly giving the decay of a signal at such high frequencies in the auditory steady state response, I think, you know, I think focusing on lower frequencies is probably more sensible. So I think in the end, it depends on the paradigms that you're using. I think we sometimes also do see activity above 60 or 70 hertz, especially in visual paradigms. You can, of course, also record resting state activity, but I think there it's sometimes somewhat difficult to interpret the meaning of this very high frequency activity. I'm also thinking, of course, of ripple activity, which some people have demonstrated in MEG, and that is, of course, maybe also of significant interest in schizophrenia in terms of learning impairments. And I think some work is currently being conducted at Oxford University on this.
Our next question, are EI balance impairments specific to schizophrenia?
No, they're not. So we find that certain abnormalities are, for example, also present in autism spectrum disorders. Generally, EI balance impairments is a very generic term, I would say. So I think it's important that, you know, more specific aspects of circuit dysfunctions are delineated. So a very general notion, they're not specific to schizophrenia. And I think, yeah, we have to, you know, be more specific about the precise mechanisms that give rise to certain deficits in order to really provide a meaningful approach, I think, in trying to decipher these circuit deficits.
Our next question, for which other disorders is there evidence for MEG constituting a potential biomarker?
So I think people are trying in several studies currently. I think Fernando Maisto, also people at Cambridge, to use MEG in the early detection and diagnosis of Alzheimer's disease and other dementias. So probably that is the second sort of major application. And I think it remains to be seen whether MEG might be more sensitive towards detecting early stages of dementias. But if, of course, that were to be true, then that would be an important case for applying MEG more widely.
We haven't got any more questions at the moment. Are there any questions before we go? It doesn't look like it. So we'll probably leave it there. Just check one more time. Yeah, I think that's all for today. So we've come to the end of our Q&A session now. Thank you, Professor Uhlhaas, for your time today and presenting this excellent masterclass session. To all of our attendees, thank you for joining us, and we hope to see you again soon. Goodbye.